Hi everyone, I am Shanidho Chaudhary of class 6 of our reputed school, Aurobindo Pathabhavan. Let's do an experiment on air pressure with an candle, water, tissue paper and all other ingredients. Now let's start. At first I am going to soak this tissue paper in this water, bowl of water, like this. After that, I am going to place this tissue paper in the middle of this tray, steel tray. Now, I am going to place this candle in the middle of this tissue paper. Now, there's one thing to understand. To lift this tray, we have to focus on the center of gravity of this tray, which is in the center of this tray, circular tray. Now, I am going to light this candle up with matches. In this way. Now as this candle gets inflamed, I am going to cover this candle up with this Pyrex glass. Now as the air inside this glass containing oxygen gets reduced, so the oxygen gets depleted, and to fill its slot the water gets raised up. Now I am going to place these glasses around this middle glass. Now I am going to show you that even if I try to lift this tray up with this glass, it's gonna be lifted up like this. See, this is one kind of, we can say, the bliss of science which we use in our everyday life. This is one of those such examples. So, I hope that you all had fun with this experiment. Thank you.
Good evening to all my respected teachers and our respected principal ma'am and all our viewers. So, today I am going to do an experiment to prove that how density acts on an object. So, for this I have taken a glass full of water, and this is actually a salt solution. And now, not a salt solution, this is, yes, half salt solution. Now I will add some more salt in it. So now it is a salt solution. Here we go. And we have taken an egg and a spoon. So, first of all, what we are going to do is we are going to take an egg and give this on the normal water, glass of water. So now, if you give it, you can see this egg totally sinks in the normal glass of water. You can see it will not float at all. It will sink. Now, we will take that egg and we will give this on the salt solution that we made. Here you can see right here that the egg is floating in this salt solution. It is not getting sinked just like this normal water that we did. It did. It is because that the salt water is more denser than the normal water here. It is because we added some salt particles which helped this normal glass of water be more denser, and that's why this egg is floating right here. and if I put this here, it will not float at all because it is not enough denser to float this egg right here. You see? There we go. Now if it is not denser, more denser, to prevent this egg from floating, I'm going to show you now. Come with me. And now we are giving half of this solution in this glass. That will be enough. And giving some normal water here. Yes. Now I'll take that egg and I will put into this solution right here. Now see, still is floating, which means that it has still denser left. So now we can do another thing. We can give more water here and we can add more normal water. And give this egg inside this. Now you can see it's acting like the normal glass of water that we did in this glass of water which had the normal water. So now why it is acting like that? Because the density is not totally gone, it is partially there, but it is not preventing it has not that enough power to float this egg inside it. You can see it's sinking. It will now not float the egg because it has lost that power to float this egg. So that was how density acts on an object. So that was my experiment. I hope you like it and enjoyed it. Thank you.
Hello everyone, I am Ritoban Swai of class 7. I read in Aurobindo Pathabhavan English Medium. Today I am going to show you how to make litmus paper at home using rose petals. So this is the litmus paper I made. Now as you can see here, there is still a white part which I haven't shaded with the rose petals. So I am shading it right now. Okay, now I am going to test it. So I will cut a part of it like this. Here. So now here is the bases, baking soda and detergent. Now with these earbuds I am going to drop the baking soda in the litmus paper. So as you can see, it turned blue. Here is the neutral part. This is a base, so it will always turn blue, as you know. And now I am removing these bowls and I will put the acids down. This is lemon juice, this is citric acid, and this is vinegar, which is acetic acid. I am not going to use this earbud as I drop the bases. Okay, so let's test this out. Wait, I have to cut the litmus paper. Okay, and now I will drop it in here. So as you can see, it turned red because these are acids. So this is how you make litmus paper at home. Thank you. Alright everyone, so now I am going to test out the red and the blue litmus papers which I brought from market. So I will tear one out. Just wait a second. Okay, so this is red litmus paper. So as you know, the red litmus paper turns blue as it comes in contact with the bases. So I will dip this thing into the bowl of detergent. And the bowl of baking soda. So as you can see, it turned blue. Now I will test the blue ones. So now here is the lime juice. I will put one blue litmus paper in this thing. As you can see right here, it turned red.
Good afternoon all. My name is Shankalpa Chakraborty. I am from class 7 and today I am going to do a science experiment. It is generally called Bush bottle rocket experiment. So now let's begin. Here I have a plastic bottle and I have done a small hole in the cap, and I have a normal perfume. So at first I am going to put some perfume in the bottle and shake it. Okay, done. Now I'm just closing the lid tightly. Now I'm just keeping this in the floor. Now take some matches, candle, I'm just directly doing it with the matches. Just put the match here. As you can see, the bottle behaved like a rocket. This happens because, as you know, in the perfume we have some small amount of methanol alcohol, which is very much flammable. So methanol is a very much has a very low boiling point. So when we put the spray in the bottle, it forms a volatile mixture of air and uh, methanol. But and when I ignite the mixture, it the high the vapor being heavier than the air, it goes upward, and when I ignite it, it forms the full air ignites, and it forms a very much high pressure zone inside the bottle, and due to the small hole present in the cap, all the full pressure wants to come out from through that one, resulting in enormous force, which is applied in backwards due to Newton's third law of motion, which we know is every reaction has opposite and every action has opposite and same reaction, due to which the bottle behaves like a rocket. Thank you.
Hello everyone, my friend Shumeda discussed the digestion of mouth, stomach and small intestine. Now let's talk about the digestion of the large intestine. First of all, I want to tell you that large intestine consists of three parts. Colon, this portion is called cecum and this region is called the rectum. The functions of large intestine are it absorbs water, minerals, and certain drugs. It also secretes mucus, which helps in adhering the waste particles together and lubricate it for an easy passage. Do you know that vitamin K and vitamin B complex are manufactured by bacteria in the large intestine? Let's come to the absorption of food. The process in which the products of digestion are taken into the bloodstream is known as the absorption of food. In this small intestine, the digested food is absorbed mainly. The absorption surface is provided with some small finger-like structures known as villi. Each villus is covered by an epithelium and has blood vessels and lymph vessels. In villi, carbohydrate, peptides, and amino acids are absorbed either by diffusion or by active transports into the blood capillaries. Heptile portal vein, which is formed by the convergence of these blood capillaries, delivers the absorbed food to the liver. In the small intestine, water, glycerol, amino acids, and some inorganic salts and vitamins get absorbed. Let's move on to assimilation. The absorbed substances are finally reach the tissues which utilize them for various activities. This process is known as assimilation. Assimilation occurs under three ways. The monosaccharides and amino acids are transported into the bloodstream surrounding the small intestine. Glucose from glycogen is transported by blood to all parts of the body. The amino acids utilized to synthesize to form new proteins which are required by the body. Excess amino acid is deaminated in the liver as it cannot be stored. The undigested food finally passes to the rectum temporarily. The ejection of fecal matter to the outside through the anal opening is known as defecation. The feces are passed to the anus by the sphincter muscles. It is a voluntary process and is carried out by a mass of peristaltic movement. So this was the whole process of the digestive system. Thank you for being with us all the while. Stay safe.
Happy Saraswati Puja. I am Sunedha Rai and today for the science exhibition I am going to discuss the digestive system. We need the nutrients present in the food for growth, energy, etc. But the problem is that the nutrients are present in such a complex form that it cannot be easily absorbed by the blood. So it needs to be broken into simpler substances. And digestion does the exact same thing. It breaks the food material into simpler substances with the help of enzymes so that it can be easily absorbed by the blood. Digestion begins in the mouth. When we chew our food, mastication occurs, which is a form of mechanical digestion. And the food is mixed with the saliva, which is secreted by the salivary glands. The salivary amylase present in the saliva acts upon the starch on the food to convert it into maltose. After the completion of the digestion in the mouth, the bolus is passed into the stomach. The gastrical glands in the stomach secretes two chemicals, pepsinogen and ACL. Pepsinogen is in an inactive state. It is acted over by ACL to convert it into pepsin, which is the active state. Proteins are acted over by this pepsin to convert it into simpler substances, peptones and proteoses. The gastric glands secrete one more enzyme, which is the renin. It is generally found in infants. It acts over the casein, which is a milk protein, to convert it into paracasein, a simpler form. After the completion of digestion in stomach, the food materials are passed into the small intestine where the digestion is fully completed. In the small intestine, the enzymes which are there are mainly produced by the liver and the pancreas. The proteins, peptones and proteoses are acted over by trypsin, which is the activated form of trypsinogen, to convert them into peptides. The peptides are further acted over by the peptidase to convert them into amino acids. Maltose are acted over by maltase to convert them into glucose. Lactose by lactase to convert them into glucose plus galactose. Sucrose, which is a milk protein, to convert it into paracasein, a simpler form. After the completion of digestion in stomach, the food materials are passed into the small intestine where the digestion is fully completed. In the small intestine, the enzymes which are there are mainly produced by the liver and the pancreas. The proteins, peptones and proteoses are acted over by trypsin, which is the activated form of trypsinogen, to convert them into peptides. The peptides are further acted over by the peptidase to convert them into amino acids. Maltose are acted over by maltase to convert them into glucose. Lactose by lactase to convert them into glucose plus galactose. Sucrose acted over by sucrase to break it into glucose plus fructose. Fats are acted over by the bile enzyme from the liver to convert it into emulsified fats, which are further acted over by lipase to convert it into fatty acids plus glycerol. All these end products from amino acids, glucose plus galactose, glucose plus fructose, emulsifies fatty acids plus glycerol are easily absorbed by the blood. So the digestion is more or less completed here. But one more step is left for those materials which are not digested till now, for the waste materials and for a little amount of water which takes place in the large intestine. This digestion.
Hello everyone, I am Anurag Kaur of class 9 and today I am going to show my sanitizer dispenser model for the science exhibition. To do this, we need a water pump which uses positive displacement principle. Here the water enters through the suction pipe and leaves through the delivery pipe. The rotating impeller pushes out the air, creating a partial vacuum, which is filled by water, and this cycle continues. Here the water enters through this pipe and leaves through this pipe. The DC motor rotates on being connected to a battery, which rotates the impeller connected to the motor. This setup is used as a water pump. Make sure that the water pump is watertight or else it may not work. The two wires, one carrying positive charge and the other carrying negative charge, connects the battery to the DC motor. But the positive charge carrying wire is separated at a point. There is an elevated base above it which goes up after pushing downwards, which is made by a connection of rubber bands. It contains an aluminium foil at its bottom face. So when pushed downwards, current can pass through it and thus it completes the circuit. As the current flows, the DC motor starts rotating the impeller. This, in turn, will take the sanitizer from the bottle and push it through the pipe. Thus, the sanitizer will eventually fall on our hand. As soon as we get enough sanitizer, we can just lift our hand as the aluminum foil is elevated upwards. Thus, the circuit will be incomplete again and no sanitizer will flow further. This is the basic mechanism of the sanitizer dispenser model. As we can see, on pushing the base, the sanitizer falls, and on lifting the hand, the flow of sanitizer stops.
Good morning, respected teachers and my friends. I am Himika Pandit from class 8 of Aravinda Pathabhavan. So today for the exhibition, I need baking soda, vinegar and a candle, and we also need a glass. So firstly, we have to put some baking soda on the glass and pour vinegar to it. See, it glows up. This is because the vinegar, which is acetic acid, and baking soda NaCO3 will release carbon dioxide, which does not support combustion. As a result, the candle just doesn't glow. So thank you. Happy Saraswati Puja.
Hello everyone, I am Pragyapal and myself Brotoja Majumdar. We read in class 9 in Aurobindo Padbhavan English medium. Today we have tried the blue bottle experiment using the following ingredients. An empty bottle with a can, sodium hydroxide solution, glucose, and this is methylene blue. First we need to pour glucose in the empty bottle. Then pour few drops of sodium hydroxide solution in the bottle. Lastly, add some drops of methylene blue in the solution. Now close the cap tightly and mix it well. Then shake it and we will notice that the solution turns blue. This is basically a redox reaction where glucose in the presence of an alkaline medium, that is sodium hydroxide, turns the blue solution into leucomethylene blue, that is colorless. This is a reversible process which we can repeat over 45 times. Stirring helps the oxygen to come in contact with the solution and hence it gets oxidized. Again, it becomes colorless when the glucose absorbs all the oxygen and it becomes reduced. After a few seconds, we will notice that the solution turns transparent. It is an example where you can observe oxidation and reduction happening simultaneously, and this is reversible. You can see here the solution turns amazingly colorless. It's totally white, and you can see a magic now that if we shake it again, it will again become blue. See, the solution have again turned blue. Now if we keep the solution again for few period, it will again turn colorless because it is becoming reduced. So this was a trivial attempt from our side. Hope you like it. Thank you.
My name is Shagnik Bhattacharjee and I read in class 8. My project mainly shows an approach for generating hydrogen which can even be used as a green fuel in internal combustion engines. Hydrogen is the first element which is found in the periodic table of elements, and it is the most abundantly found chemical substance in the universe. And as we can see, it has a very high calorific value, which makes it a very efficient fuel for our daily use. It can either be used as a fuel cell or in internal combustion engines, but the cost of producing hydrogen is very expensive. Whereas we can generate a hydrogen-Nal gas mixture by using the very simple process of electrolysis domestically in a very cost-effective way. And if we get to use any solar cells for generating the electrical energy for electrolysis, then the process becomes totally free of cost. Now, because of the lack of resources, I have only used a plastic bottle, some steel plates, and an aqueous solution of NaCl and sodium bicarbonate for the electrolyte. And by passing an electric current of about 1 ampere, I have made the following setup. So this is a sanitizer bottle which is acting as the container, and it has been made leak proof. So this black wire is connected to the cathode and the red one is connected to the anode, and the pipe is attached to it for the mixture of the gases to come out. And this is having aqueous solution of NaCl. The electrodes are unreactive for not letting any unwanted reaction to occur. So when electricity is passed, there is an excess of electrons in the cathode and deficiency of electrons in the anode. And by this gaining and losing of electrons, some chemical reactions will occur which will produce the gases. But before going there, we need to understand that this may look simple, but when a current is passed, by Kirchhoff's voltage law, we can conclude that about 8 volts of electricity is wasted as heat, and which can be overcome by placing a neutral plate in between the electrodes, thus reducing the wastage of electricity by 4 volts. Now, as the NaCl molecules are already being dissociated into Na plus and Cl minus ions by dissolving in water, so when the current is passed, Na plus and H plus cations start migrating towards the cathode. The Cl minus and OH minus ions start migrating towards the anode. The H plus ion receiving the electron more easily than an Na plus ion becomes an hydrogen atom. On the other hand, hydroxyl ion by losing electrons become two H2O molecules and one oxygen molecule. Now the ratio of the volume of the hydrogen produced to the volume of the oxygen produced is 2:1. Moreover, theoretically, this ratio of the mixture is able to gain its maximum efficiency and is an extremely explosive mixture, which can obviously be used as a green fuel or can be used for enhancing the combustibility of other fuels. And here is a demo showing how much combustible this hydrogen NAL gas mixture can be. The following demo has been conducted under the supervision of a teacher, and no one should try to replicate this because it's dangerous. So that's it. Thank you.